15 seconds or so.
Okay.
Hello, welcome back to our discovery track. Thank you for joining us again. And our speaker for this session is going to be Fiu Zin. She's going to talk to us about how we leverage automation for data mining, pre-processing, and curation. And I think this follows really nicely on the book chapter that was written by Bob and Pankaj about good practice in model building that brings some automation aspects to it. So, Fiu is a scientist at Simulations Plus. I think we could call you our scripting wizard, and we certainly look forward to hearing about this topic.
Thank you so much, Eric, for the introduction, and thank you everyone for being here. So in today's presentation, I'm going to be covering how we leverage automation for data mining, pre-processing, and curation. So to give a general overview of our cheminformatics machine learning project, the very first step is data mining. This is where we collect data from commercial public databases and literature. And the next step is data pre-processing. So most of the time when we have collected the data, they are usually very noisy, messy, and contain a lot of irrelevant information and can also come in different formats. So we deal with those complications in this step and then transform the raw data into a useful, efficient format. And the next step is data curation. So in this step, we ensure that the data set is high quality, making sure that it contains biologically relevant information for the endpoint that we are interested in modeling. And we do a lot of mistake diagnosis and correction in this stage because there are usually a lot of mistakes present in those databases. Next, we calculate molecular or atomic descriptors and do feature engineering as needed. And then we split the data into train and test sets, sometimes using random splitting or cohort mapping. And then we choose a machine learning technique, sometimes a simple vector machine or artificial neural network. And then we start building models and validating them. So based on whether it is a classification or regression problem, we choose a proper set of evaluation metrics and select some of the best models. And then we assess the applicability domain of our models. So among all the steps that I have described, the very first 3 steps, which have to do with data handling, is usually the most cumbersome and expensive of all, and they are usually the most heavy lifting part of the entire pipeline. If we look at it, normally data mining, pre-processing, and curation take about 85% of the work and the project timeline. So to deal with those, we often use automation when dealing with those processes. And the major thing is that we wanted to overcome some of the most tedious and repetitive and time-consuming tasks for humans. We wanted to also ease the strain and burdens on humans. And we use automation in a way where we can improve the quality of our work and not only efficiency, and also minimize the human error. For example, if we were to do it manually, that would introduce a lot more mistakes than needed. So we try to use automation to overcome that. And we use automation to address common problems in the pipeline that has a clear, well-defined set of rules. So here, the bubbles that are popping up are some of the example cases where we use automation. For example, we use automation for structural retrieval, rule-based information mining, text mining, handling duplicates, and flagging suspicious entries, and so on. So I'm going to show 3 example use cases of automation from one of the projects that we have worked on, which is the cytochrome P450 cytometabolism project. So the very first example that I would like to show is how we use automation to do web scraping for drugs from the FDA website. So if you go to the FDA website, they have approvable drugs listed and categorized by year. So they have these tables on their page, along with some other information at the top. But these are the tables that we are interested in extracting. So given a range of year, we extract all these tables and information inside. But not only that, each drug is hyperlinked. So if we click on that, it goes to the next page. And it has a lot more additional information that are relevant to this drug. And we are specifically looking for this label PDF document because it contains a lot of relevant information to our project. It has CYP metabolism information and perhaps drug-drug interactions. It has a lot of relevant information. And if you click on that, it opens the next page, which is searchable. So we have a predefined list of keywords. So we know the kind of CYP enzymes that we're targeting. For example, we see that here is 2C9. This is one of the important CYP enzymes that we're going to be looking at. So we have a list of keywords, and we use them to do the text search for this entire PDF. We do this for all the drugs. And then if those keywords are present, we collect those keywords and then assess the priority. So if many of them are present, we consider that a high priority drug. So all the steps that I have mentioned here are entire automated in the pipeline. So we have an output like this. So we have all the drugs here, and along with the links that is for label PDF and the keywords detected in each article and the priority assessment. So we focus our literature mining efforts to high priority drugs because we know that there is a lot more information out there for CYP metabolism and metabolism schemes. And it saves a lot of time, effort, and headache that would otherwise be devoted to low priority drug. Because usually for those compounds, CYP information is incredibly hard to find and challenging to find. So that would eventually be dead ends. So we try to be efficient at which drugs that we're going to be targeting first. So in this example, I show that this automation pipeline efficiently mines hundreds of drugs and then help us prioritize them for the cytometabolism project. And this whole process is completed in just minutes. The next example case that I want to show is how we use automation for structural extraction and verification. We use it very often because in a lot of the data sets that we have chemical names, but we don't have structures. And sometimes we have structures, but there are a lot of mistakes present in those structures. So the way that it works, we feed a list of chemical names into the pipeline. So let's start with one example. Let's say there is this name that we are trying to look for structures. So we use backend APIs and sometimes web scraping from multiple databases out there. Based on the name, we extract the structures. And all these structures go through chemical standardization. For example, generating canonical smiles, standardizing tautomers, salt removal, and so on. They go through all these processes. And at the end, after they have been processed in the same way, they are compared. So based on the comparison results, there are 4 categories. And in this case, these are all the same. So it goes into agree category. But if the results are different, then they are going to go into suspicious entry category, because likely that one of the 1 or 2 databases have reported wrong structures. And if we only have structures from only 1 or 2 databases and we have no other information from other databases, we put them into not enough category. And if there is no structure at all, then we put them into nothing here category. And most of the time, the effort that goes into, most of the effort go into these 3 categories, because these are the problematic ones. So we have to actually dig into literature and find out what is the issue with them. And sometimes there are mistakes in the name, so we need to do additional runs for this automation pipeline as well. So just to show how it kind of looks, here is a list of chemical names that you will feed into the pipeline. And along with the other structures and IDs, we would extract all of them. And then there's the column that kind of summarizes the results. And then we focus most of our efforts on the suspicious and the other categories I have mentioned before. So to summarize this case, we use this automation pipeline to extract thousands and thousands of structures and verify them. And it can be used across any cheminformatics project. And we use it in a way that can improve the data quality. And we have used it so often that is now an integral part of our data curation pipeline. The next case that I would like to show is how we use automation for margin data sets. And of course, the way that we march and format data is different for each project. And the example that I'm going to be showing is very specific to cytometabolism project. So here are 3 example databases, and we have already formatted them to have a similar format. So let's look at the database one. So we have a database one, a screenshot of database one. It looks like that we have structures, identifiers, and smiles, and we have CYP columns here. So these are for individual CYP enzymes, and these are the atomic sites of metabolism. So we have the numbers here. And you will also see that some of the formats are kind of odd. There are 2 columns here. So we also account for them as part of the automation pipeline. The problem is when we merge these data sets from multiple databases, then there are going to be a lot of conflicting information and entries. So how do we deal with that? So these are a set of rules that we have in mind, especially one handling duplicate structures for margin datasets. So we'll march all the sites of metabolism for each SIP and information in all the other columns. And we also make sure that the delimiters are consistent, and there are going to be several IDs coming from different databases. So we put all of them in the alternate column. We keep only the first one in the primary ID column. And then we flag all the suspicious SIPs that will need eventual, manual checking. So to look at an example, I'm simplifying it, showing two examples coming from two different databases. This is database 1, database 3. And these are duplicate structures based on the canonical smiles. And if you look at them, there are going to be conflicting information. For example, SIP 2A6 and 2B6. It's likely that one of them is wrong, or they are not properly reporting. They may be missing information. So we don't know that, but these definitely need to be checked. So after merging them, let's see. So we have this canonical smile. This is the same, so it doesn't matter. So the second one, which is the ID, it goes into this alternate name column. We have merged all the references. We have merged all the data sets. And we have also merged these as well and sorted them and properly put commas to separate them. But we also need to account for them. We need to go back and check that. So we added an additional column, suspicious column here. So essentially, whenever the information is conflicting, we track all this SIP enzymes information here. So at the end of the marching, we just go and check these columns and make sure to dig into the literature and properly update them and do corrections as needed. And if you think about it, these are just two examples that I'm showing here. There are going to be a lot of multiple duplicates coming from multiple databases, and it's going to be painful to have to deal with them manually. So the tasks involved in this data merging process can become very redundant and also very time consuming. And it's so easy to make mistakes, especially when we're looking at hundreds of numbers like this. And it doesn't make sense to do it manually. So here, this automation pipeline merges all these multiple data sets in just a few seconds, and while following all the consistent rules that we have coded and dealing with all these complications in this process. So to conclude, I have shown 3 example use cases of automation from our cyto metabolism project to highlight the efficiency, usefulness, and importance of automation. And we use automation often in data mining, preprocessing, curation, mainly to overcome very repetitive, exhausting, and time-consuming tasks, and also to ease the strain and burden on humans because it can take a toll. Also, we try to minimize human mistakes, manual errors. If we were to do it manually, that would introduce a lot more errors. So we use automation to overcome that. And we use automation in a way to enhance data quality and also to improve efficiency by lowering time, cost, and effort. Last but not least, I would like to acknowledge my team members, Pankaj, for all his help and support for all my projects, and the rest of my team members, Michael, Bob, David, Marv, Robert, Vieira, Eric, and John. I would also like to acknowledge my cat for his contributions in my project. He's usually sitting around on my desk and giving moral support. So thank you, Kat, for your company and attention as well. So thank you to the audience.
Thank you for your very interesting work. So I think we have about a minute and a half left or so. There's just enough for me to launch the last poll. I'll just do that right now. If you can just tell us what you thought about this kind of automation work. Maybe this is something you're already doing, and see that have deployed this kind of semi-automated approach, really, because we're just trying to focus the human effort on the most relevant part, which is the really interesting aspect. All right. Well, with less than a minute to spare, I think I will retrieve the poll, and it looks like almost 80% of our attendees thought that was really cool stuff. So thank you for your contribution, and I'm going to close the session now, and then we'll see you in the next talk with Marvin Wallman.
Thank
you.
Thank you. Bye-bye.
Bye.